Hello, everybody. Getting ready. So this is, I guess, our third lecture in neuroanatomy. And we first two lectures were at the microscopic level. The next two lectures are going to be not just macroscopic, will be at the gross anatomy level. Today, we're going to look at the cerebral hemispheres, so telencephalon. And next week, we will look at the lower portions of posterior fossa, basically, and some of their upper diencephalic structures. So let's just start. Here's our outline. We're going to be looking, we're going to have, this is also a bit of an introduction, so we're going to look at the major divisions of the nervous system and some anatomic terminology. And then the main focus is going to be on the cerebral hemisphere's gross anatomy. And this is fun. This is just looking. This is all about looking. Major divisions, you have, so the nervous system consists of the central nervous system, a peripheral nervous system, and an autonomic nervous system. This course is only about the central nervous system. The central nervous system consists of the brain and spinal cord. And at least for the next two lectures, we're going to be looking only at the brain. We will get to spine later. The brain, I think, is conveniently divided up into cerebral hemispheres, the brainstem, and the cerebellum. And today we're only talking about the cerebral hemispheres, but that will include the basal ganglia. And throughout the course, and certainly today, you're going to see the importance of embryology and development, starting right now. So we're going to have, there will be lectures on embryology, and we will get to lots of detail. But for today, just as an introduction to today, you can see that at the 4 weeks of gestation, the fetus, the developing embryo, has three primary vesicles in the nervous system. And I think this really points out how this notion of tube and the wall really is really key in the early development. And that this three vesicle stage, we have the forebrain, which is the prosencephalon, or forebrain, the midbrain, which is the mesencephalon, again midbrain, and the rhombencephalon, the hindbrain. And then a week later in gestation, you're up to the 5 vesicle stage, where now the telencephalon, or the prosencephalon, has separated itself into the telencephalon and the diencephalon. And very much today, we're going to focus on the telencephalon. The midbrain stays as the midbrain, and then the hindbrain, the rhombencephalon, will split another new vesicle into the metencephalon and the myelencephalon. And these will be the medulla and the pons. And you can see what their adult structures are over here. And like I said, we will spend more time on this later. Just wanted to show you some of the, here's an embryo, a human embryo. And these are the 3 planes. Okay, let me introduce a new concept. From this talk on, I'm going to expect you to spend a minimum of 1 hour a week outside of this lecture studying the material in this lecture. So you can pull up the lecture, you can go to references if you want, although I don't think you'll need them. But I would like you to spend a minimum of 1 hour a week going over every lecture that we do from now on. If you don't, you're not going to be able to keep up and you're not going to benefit, because I cannot possibly verbalize every piece of anatomy that we're going to be covering in these lectures. You really, as you're going to see in a minute, you're going to have to just go over them and you don't need to write things down. You just need to look at the slides and look at the slides, okay? At any rate, these are just the pictures on the different planes that are here of the diencephalon. This would be the ventral third ventricle. Here's the telencephalon. These will be the lateral bodies of the ventricles. You can see here that this is actually the rhombencephalon. And you go, whoa, why isn't that the telencephalon? And because, well, look at the plane, you see, and it's going through the rhombencephalon here. And here you have, again, rhombencephalon in a different plane. This is this plane. And so you can see. And then this is the only one, the slice 1 is the only one that runs from the telencephalon through the diencephalon and then into the rhombencephalon, which is the largest ventricle at this stage. Anatomic terms. Here's the issue. In the spinal cord, and for most animals that exist, the term dorsal and ventral are perfectly adequate. But there's a flexion that occurs in the human nervous system and other mammals that makes dorsal and ventral become almost ambiguous. So this now it remains dorsal, strictly speaking, and ventral, strictly speaking. But you have this new dimension then. If you're going to use it, if you're going to use dorsal and ventral for that, then you're going to need rostral and caudal for another dimension. Okay. And also remember that what was dorsal is going sort of side to side is no longer. All right. In here in these pictures with this woman, so you can see our classic anatomic planes, the axial plane, the coronal plane, and the sagittal plane. And we use that kind of information all the time. All right. Let's look at the base of the skull, which holds of course, holds the brain and is our guidelines for the cerebral hemispheres, especially. You know most of these, so I just want to point out here the petrous bone and the sphenoid. And if you ever are in doubt about what the sphenoid is, think of this picture and the term sphenoid wings. Okay, it's almost like a butterfly. And when you look at it straight down on it, and the large petrous bone, which is also fused with the temporal bone. Okay, but I think this picture is also helpful because it shows you the tentorium and the falx. And you can see how the falx and the tentorium divide up the skull into the right and left sides of the hemispheres, and then also separate the posterior fossa from the middle fossa, okay, and the anterior fossa. So anterior fossa here, middle fossa here, posterior fossa here. It's funny, we use these terms like posterior fossa. We don't think of, well, other fossa exist, but they do. All right, let's start looking at the brain, and we're going to use the same images over and over, and there are a lot of things for you to look at, but we're just going to focus on what this is telling you to look at. This title tells you all that this is what I'm expecting you to see off this picture, and we just want to, in this view, we just want to highlight the interhemisphere fissure. And that's basically it. We're going to get to these orbital surfaces of the frontal lobe shortly. And this is, of course, where the falx cerebri sit. All right. And we're going to just keep going. We'll come back to that. You then we're looking at the major fissures. You have the Rolandic fissure and the Sylvian fissure, or the central sulcus, central fissure, whichever you want to call it. These are your guidelines, right, that are going to help us divide them up into various lobes, all right? This here, so here is your Sylvian fissure, and this is the Rolandic fissure. All right, this is everything rostral to this, I'm going to say, anterior, if you like. Anterior to this is going to be the frontal lobe. All right, here's the Sylvian fissure. This is going to also separate frontal lobe from temporal lobe. Parietal lobe is going to be back here, and occipital lobe. We will look at all of this. And we're not looking at the posterior fossa. We don't want to look below the tentorium today. We're going to get back to all of these gyri in a little bit. The main thing right now is just to pick up these major fissures. The insula is hidden. And the insula also is related to the basal ganglia, really the putamen and the caudate. We're going to talk a little bit about the basal ganglia in a couple of minutes. And there is no real, in my mind, there is no limbic lobe. The limbic is a system. It is a concept that is put together and varies from time to time. It's not easy to define anatomically. Other people would say, well, it is, but for instance, here's the transverse temporal gyri, and these are the long insular gyri. So is this temporal lobe, or is this temporal, or this is really all insula. It's a series of very different gyri that derive from the same tissue as the putamen. Again, we'll come back to all that in later detail. But this is exposed when you pull down on the temporal lobe, as it's seen here. All right, as we start to look at the more detail of the gross anatomy of the cerebral hemispheres, you really have to think about Brodmann's map, right? And Brodmann's, the concept of Brodmann's map, Brodmann wrote this in 1908. Someone asked me a few years ago, if isn't Brodmann's map disappearing? And my answer was, on the contrary, everything that we think about in the cerebral hemispheres is derived from Brodmann's map. And all of the gorgeous growth of anatomy, growth of embryology, growth of physiology, all really continues to strengthen the concept of Brodmann's map. It is central. And basically what Brodmann did is he just sampled lots and lots of cores of tissue of the cortex all over the cerebral hemispheres. And he kept great detail of the map. And wherever he saw a transition of the anatomy, he just put a little boundary. And eventually he had these boundaries all over the place. And then he stepped back and looked at what he had created with these boundaries. So this is all about transitional boundaries. Transitions of what? So what Brodmann did was he, first of all, used the developing idea that was very current at the time, that modern cortex is a six-layered cortex, that's neocortex. And that the 6 layers, there was a pretty standard, that 1 was a molecular layer, and 2 tended to be a small cell layer, and 3, if there was a 3, tended to be a pyramidal cell layer. Then came another granule cell layer, then came another pyramidal cell layer, and then came this area that is of spindle-shaped cells with very different cells. So he said, good, all right, we have 6 layers. He took that idea, and then what he saw was that from one extreme to the other, look at this, he found areas such as the precentral gyrus, all right, the precentral gyrus had very few of these small granular type cells. Layers 2 and 4 were minimal, and layers 3 and 5 were everything. All right, and always some of these spindle-shaped cells making layer. But note 2 and 4, and this is 3 and 5, and this eventually gave rise to the corticospinal tract. So he called this a type 1 cortex, which was all output neurons, all these big pyramidal neurons, which are output neurons. At the other extreme were the primary sensory cortices, and the calcarine cortex would be a good example, where layers 3 and 5 are nearly non-existent. And since there's almost no layer 3, 2 and 4 almost merge with one another. And this is the type of primary sensory cortex. All right. And so somatosensory, Brodmann's area 3, 1, and 2, Brodmann's area 17, Brodmann's area 41 and 42 for the auditory. Each of them has a structure of almost pure granular cell, 2 and 4 level, minimal 3 and 5. And then surrounding, as you move forward, in this case, from the primary motor strip, you see a secondary motor area, which is mostly level 3 and 5, but now you're seeing a reasonable amount of 2 and 4. This will become Brodmann's area 6 and Brodmann's area 8, which will become the premotor and supplementary motor cortices. Coming from the other direction, you start to see pyramidal cells. So now some projection stuff is starting to develop into this primary receptor. These will become Brodmann's area 8, 18, and 19, Brodmann's area 5 and 7, Brodmann's area, all the temporal gyri that we're going to look at in a little bit. And finally, Brodmann recognized that there was this very balanced cortex where you had seemingly equal weight being given to the receptive granular cell layers and the projective pyramidal cell layers. And he called these the association cortices. These are the types of cortices that we're going to eventually get to when we get to our late lectures in this course, that we're looking at association, that these are going to become involved in working memory and concept of language and decision-making and perception of the world. Okay, and so then other people have built on this so that while you have all of these Brodmann numbers, This is a nice color scheme, and I will use this many, many times in this course, where blue is primary. Yellow is, and it could be primary motor or primary sensory. Yellow is secondary. It remains relatively modality specific, supporting and enhancing the complex analysis of the primary data that's coming in and projecting it elsewhere. And then this all of this salmon colored stuff, okay, these are your association cortices. All right, this is frontal lobe, prefrontal cortex. Okay, all your decision making. Okay, these are your language centers. Okay, your interpretation of objects, facial recognition. We're going to look at some of the lobes that are involved in that. And same thing present on the medial surface. And finally, then, just to complete this, you then have the atypical limbic cortices, often three-layered parahippocampal, the temporal pole, the cingulate, and we're going to look at those in a little bit. That becomes, and the olfactory interpretation cortices, these are going to become your limbic system. Okay, now let's look at frontal lobe. And so we're going to look at similar pictures, but now we're going to look at the frontal lobe in these pictures. Again, this whole lecture is just looking. The precentral gyrus is the posterior limit of the frontal lobe. You have three horizontal convolutions, the superior, middle, and inferior, not surprisingly. And the inferior is of great interest to us. I would also say the gyri are only loosely associated with Brodmann's map. Brodmann's map maps to function. The gyri, not so much, okay? But this inferior one is interesting because it contains the pars triangularis and the pars opercularis, which are Broca's area, and so there is some sense to that. So we're going to look at so this is postcentral gyrus. We are not interested right now. Don't look at that. Okay, here's so here's a Rolandic fissure. We're interested in everything forward of that. There's your precentral gyrus, and then you have superior, middle, and inferior transverse frontal gyri. Okay, and then we don't look at the orbital. We'll come to that later. And then here's your pars triangularis. That's easy. Okay, and this is the pars opercularis. What does operculum mean? Why do we call it operculum? Operculum is lips. Okay, these are in the lips of the Sylvian fissure, upper lip, lower lip, right? Okay, that's the way. So you're going to have an opercular portion of the parietal lobe, which is going to be the inferior portion. You're going to have an opercular portion of the temporal lobe, which is a superior portion, because it's on the opposite sides of the lips. Okay, and then you have the frontal pars opercularis, which is part of Broca's area, and then the pars orbito frontalis, I think it's called. And here you go. And this is again just highlighting the Broca's area, the pars triangularis, and the pars opercularis. All right, orbital surface. You can see that this thing is sitting on the orbit, right? I mean, it's sitting right on the orbit. So here are the pieces that I want you to look at. Here's the gyrus rectus, straight as an arrow. We're going to look at that in another view shortly. You have the lateral orbital gyrus. Let's try to find that. The lateral orbital gyrus would be out here. Lateral orbital gyrus. So here, presumably, this is the medial orbital gyrus. It's unlabeled in this picture. And then the olfactory bulb. Just there, you can see the bulb sitting in the groove. That's all I want you to pick up out of this one. All right. Then you have we're still focused on the frontal lobe, but now we have the orbital view, and you can easily see the gyrus rectus, the olfactory bulb, the olfactory tract. Now we're getting into diencephalon, so don't look at that, and temporal lobe, and don't look at that either. So all we want to look at right now is the frontal lobe, and this is just what it looks like on the orbital surface of the frontal lobe. All right. Medial surface of the frontal lobe and the interhemispheric fissure. All right. The cingulate and the cingulate gyrus are very prominent, but we don't want to include those. They're not neocortex. They're three-layered cortex. So we're going to talk a lot about the cingulate. The cingulate is very sophisticated. Do not think because it's three-layer that it's like, we could do without this. It's not, okay? the three-layered cortex. It's extremely valuable, as we'll talk about much later in the course, in terms of assessing the success or failure of our actions, which is pretty important stuff, okay? Osler's rule, right? If it works, do it again. That's coming out of the singular, okay? But at any rate, right now, we're not going to consider it because it's part of the limbic system. And we're going to instead look at things like how the precentral and the postcentral gyri, precentral and postcentral gyri of the lateral surface of the brain on the medial surface, they fuse to make the what's called the paracentral lobule. Okay, so here we are. All right, here's the, this is where the Sylvian fissure, excuse me, the Rolandic fissure would be. And so strictly, this is frontal and this is parietal. But you can see that on the medial surface, they come together to make the paracentral lobule. I sometimes think of that as being appropriate because of what we do with our legs is so often things like walking and swimming, where both legs are involved nearly simultaneously. Anyway, here's the corpus callosum. We're not going to look at that right this second. And this is the cingulum here. All right. But, and there are other things that are of interest, but we're not going to look at those right now. Still, here's your gyrus rectus, all right? And not a lot of markings that we need to pay attention to. That superior frontal gyrus that we only saw a little bit on the lateral surface, when we looked laterally, is more visible here on the surface. Okay, let's move on from the frontal lobe to the temporal lobe. We're all just looking at surface anatomy today. The lateral surface of the temporal lobe has 3 oblique gyri, again, superior, middle, and inferior. The superior is particularly of interest because it ends as the angular gyrus of the parietal lobe. The superior surface contains the primary auditory cortex, and that is including Heschel's gyrus. Superior temporal gyrus, middle temporal gyrus, inferior temporal gyrus. And that's all that you need to see from the lateral surface right now. We put this up here. We previously looked at Broca's area. We see the primary auditory area, Brodmann's 41 and 42. And then Wernicke's area is ill-defined. There are some anatomists who would like to include all of this in Wernicke's area, and certain anatomists, and from a clinical point of view, generally we do. All right. But this is, strictly speaking, others would be more strict and say, well, Wernicke's area is the receptive area for language reception. And these areas are not really so much receptive. They have more to do with language, perception, speech production, okay? And so they sort of want to divide this up. And certainly the supramarginal gyrus is among the most important and complex structures of the brain. And so there's some justification for separating them, although, again, clinically and historically, Wernicke's area was this entire group. I'm not going to be able to get used to that. And you can see, if we look at the temporal lobe here, that Brodmann's 41 and 42 are primary. And then they're surrounded, as almost always, surrounded by areas for interpretation. And then as you move away from that into areas which are at the boundaries between vision and hearing, between vision and somatosensory, between hearing and somatosensory. At all of those boundaries, in the spaces in between on those border zones, are the association cortices, and then this mass up in the prefrontal lobe. We will talk about prefrontal, and we will talk about this at almost at the end of the course. And when we talk about this structure, the topic, the organizing topic, will be free will. We're going to talk about free will, the anatomy of free will. The temporal lobe, all right, again, on the inferior and medial surface, there's a little bit more going on, more parts to name. You have this long-running occipitotemporal gyrus, a parahippocampal gyrus. You're going to have the piriform lobe working in here, which is olfactory. This is the primary olfactory cortex of the piriform lobe, which is not easily marked. I've tried to mark it for you. This will include the uncus and the lateral olfactory stria, and the fusiform gyri, which again is, these are things that are not The fusiform actually is easier to identify. I put these things on here based on some other drawings that an anatomy, just to add these in here. So this is your piriform area, and this is the fusiform area. Very complex, crossing gyri. But let's just look for a minute. First of all, here, right, this is so let's look at what's named here, right? This is the inferior temporal gyrus, right? This is the uncus. And here on this side, we have the temporal lobe is dissected free. So we're not looking at surface anatomy, but you can see how the amygdala sits. Juan and I have been going back and forth on things like this. Do you count the uncus as an anatomic structure? It's a gross anatomic structure, but it really is where the amygdala protrudes off the parahippocampal gyrus. And you know, it's semantics, you can call it as you will. Here's the parahippocampal gyrus. Here's that big occipitotemporal gyrus here, and will become the fusiform gyrus, which we have better pictures I'm going to show you in a minute for fusiform. All right, but this is piriform in here, all right, and the fusiform. This picture is mostly not what we want, but I wanted to show you how the olfactory tracts come back and land into the olfactory trigone area. I also wanted you to see the uncus sitting here and here. All right. And here's the insular cortices. We pulled the temporal lobe away. Here's the transverse temporal gyri. And here's again, the insula. Pulled away and dissected. It's not just pulled away, but we've dissected prefrontal cortex. We're going to have the embryology is an enormous help in understanding this anatomy. It just helps because the neocortex is folded on top of everything else. All right, let's go back to the surface anatomy of the parietal lobe. You have, again, you start with the central sulcus, lambic fissure, and you come backwards. And this boundary between the parietal lobe and the occipital lobe is very imprecise. It's a little crevice in a sulcus. It's imprecise. You have the post-central gyrus, the superior parietal lobule, the inferior parietal lobule, which we've already seen as the supramarginal gyrus and the angular gyrus. And then a very interesting structure called the precuneus on the medial surface of the brain. And the precuneus is about gestalt perception. That is, you just sort of take the whole thing and go, I know what that is, or I know who that is. We'll come to that kind of idea, not in the precuneus, but lower down. And mental imagery strategies. If you were working your way through a three-dimensional puzzle, what you have in your mind as you're trying to navigate this before you get there or hit the wall, that sort of thing. I think that our interventional radiologists have a very well developed precuneus because they're seeing this before they see it, right? They're navigating without constantly repeating the angios that I would need to do, certainly. Here we are on the parietal lobe on the lateral surface. Here's the post-central gyrus. Here's the supramarginal gyrus, again, the Sylvian fissure. Supramarginal gyrus, a very large structure, really, and the angular gyrus, which sits right at the end of the Sylvian fissure, right at the angle of it. And so those are the things you want to take away from this view. On the medial surface, again, remember you have the paracentral lobule, which is fused. This is all cingulate. Here's the precuneus. We're going to get into cuneus, that's the primary receptive area for vision. There's the cuneus. Okay. And the occipital lobe is surface anatomy. This is your, of course, primary visual cortex. You have the cuneus, which is, again, that's the receptive area, and the lingula that I want to show you. Lingula is where you have these more, again, more complex visual analysis. Remember, a secondary cortices bordering into a association cortex for the highest level of analysis and finally merging with concepts so that you start to get into what is this? How is this? What letters are these? But also logical sequencing, which is interesting, and the encoding of visual memories, which is an interesting thing in itself. Not just the visual memory, but the encoding of it for storage. Okay, so cuneus, Here's the parieto-occipital sulcus. This marks the boundary of parietal from occipital. That's easy on the medial surface of the brain. Here's your cuneus. There's the calcarine fissure, right? Heading out, separating out. This is your primary visual area. Okay. And here is your lingula, which has all of this very sophisticated visual analysis, again, reading and things like that. Okay. And this was the good view. Here's the lingula that we were just looking at. And this is a color-coded picture that shows you the fusiform gyrus down there. That was hard to demonstrate on the temporal lobe there. Okay, here's a parahippocampal gyrus. Here's the entorhinal cortex. Pericalculin. We notice we're on the interhemispheric side. The pericalculin, calculin, right? Cuneus, precuneus, paracentral lobe. Okay. Dorsal surface of the brain. Not much to talk about. You're running down the interhemispheric fissure. Mostly what I want you to see is look at this. Look at this. This is the central sulcus. I think this is the first time that you really get a sense of how big the frontal lobe is. Okay. And also when you're working in the CT plane, and you want to think that the forward half is the frontal lobe and the back half is the parietal lobe. It ain't. Okay. It's not like that. Okay. It's not. The parietal lobe is relatively small compared to the frontal lobe, which is huge. It's just huge in the human. It's gigantic. Okay. Occipital pole, nice and simple. Tentorium would be sitting here. Interhemispheric fissure, right? There's your calcarine sulcus is up in here. Cuneus is going to be here, right? And then this is the lingula, all the way down here. So again, on CT scan, when you think you get down to the very base, you're not. If you're at the very base of the occipital lobe, you're not actually in the primary sensory area. You're in the lingula, which is going to be conceptual. And the patient may not have a hemianopia, but may not be able to read or recognize their brother. Okay. All right. Let's get into white matter. All right. Lots and lots to cover. All right. We're still in the cerebral hemispheres. So I want you at this level of discussion, we're going to think about 3 different types of white matter pathways. All right. There are projection pathways, association pathways, and commissural pathways. A projection pathway we will define as something that leaves the cortex. It leaves the cortex. It can go down other parts of the cerebral hemisphere. It might go down to the basal ganglia, but we call it a projection if it leaves the cortex. It could even go, leave it at that. It leaves the cortex, and it could be corticospinal too. Association fibers are fibers which go from one region of the cortex to another region of cortex in the same hemisphere. Okay, so that's an association cortex. We'll look at some, but this is conceptual. Projection leaves the cortex. Association is from cortex to cortex in the same hemisphere, and commissural is cortex to cortex in the same hemisphere. There are other commissural fibers that are not cortical, but we're going to focus today on the ones that are. And then the centrum semiovale is sort of a regional term that we will use. We always use it clinically, and so we will use it. We're going to look at the corona radiata. We're going to look at internal capsule. So now we're looking at projection pathways here. Our projection pathways are corona radiata, internal capsule. They're all the same, right? It's a projection pathway going out, okay? But they get off at different places: corticothalamic, corticopontine, corticobulbar, corticospinal, right? They travel down the brainstem as the crus cerebri, as the basis pontus, as the medullary pyramids. We're going to also look at projection pathways that are the optic radiations and the thalamocortical radiations, thalamocortical. Here you go. There's a corona radiata. I love this dissection. When I was a resident in neurology, we were the second year neurology residents were the teaching assistants in the medical student neuroanatomy course. And we would meet the night before with Len Hamer, great anatomist, and Len would do the dissection for all of us. And then we would go the next day and do the dissection for the medical students. It was wonderful. And this was my favorite dissection. You just get to peel away cortex and you hold this gorgeous fan in your hand going down and showing you its continuity as internal capsule into the basis pontous, down the pyramids. I loved it. I absolutely loved it. Now, with this slide, we're going to introduce this is the Yakovlev Halim collection. Yakovlev was a neurologist who collected specimens of brain from 1930 to 1974, an entire career. And this is his collection. Look at the size of these slides. These are whole brain glass slides. Okay, I can't remember whether it's University of Michigan or Michigan State, which has taken this collection or a similar collection, I'm not certain, and they have provided it to us in such a way that we can digitally zoom in on any segment of it. This is online, real time, and see the histology of it. So I will get that website for you. It's an awesome tool. It's awesome. And it is virtually, I'm just going to say it's an invaluable tool. And the Yakovlev Halim collection has all of the slices matched. So section 1700. All of these sections are matched with a Weigert stain for myelin and an unstained cortex, or a Nissl stain for the cell bodies of the neuron. And so together they are unbelievable. And the interactive set has both. You can switch from Weigert to Nissl, and you can zoom in and tell it what power you want anywhere on the slide. It's unbelievable, unbelievable piece of data storage. Okay, here we see, and so we're just, okay, I'm not going to get into it here. This slide, all I want you to see is here's the anterior commissure, okay, which is running across from parahippocampus. And here's the posterior commissure, which is going to be more visual in its orientation. Some language going to be in here. This is connecting the pulvinars, and the pulvinar are both visual and language. Form follows function. You're always going to find it. Look at how the basal ganglia are outlined in this. We're going to look at those in a minute. Here's the claustrum. So yeah, this is just work looking at these lower structures and how you can start to see nuclei within the thalamus, all right? And how you can see, here's medial geniculate, there's a lateral geniculate, the folium's and stuff like that. And so now let's just look, all right? We're going to look at, first of all, association pathways. You have short association pathways. They go right under the sulcus, so short to short, okay? Then you have longer that go from one lobe to the next, all right? The superior longitudinal fasciculus run from between the frontal and parietal lobe, frontal occipital lobe. Inferior longitudinal fasciculus between the parietal and occipital to temporal. The uncinate fasciculus between the frontal lobe and the temporal lobe. The arcuate fasciculus, as you know, we think about it all the time in language, frontal parietal, frontal temporal, and then the cingulate fasciculus, which runs frontal parietal connecting the parahippocampal structures temporal lobe. Again, I love these, and you can see how the cortex interacts with all these saddles and that sort of thing. And you can see here, this is a superior longitudinal fasciculus. There's the arcuate fasciculus making an arc as it's going down into the temporal lobe, right? Here's the uncinate fasciculus running in that connection between the frontal lobe and parietal. When I see the uncinate, I think about function, but I also think about glioblastomas traveling from the frontal lobe down into the temporal lobe via the uncinate. Okay, frontal occipital projections, and then the inferior longitudinal fasciculus here. Pictures, that's what I'm saying. I can't name all these structures and go over all of those. Here's the cingulum. You're just going to have to take this apart and see going down into the fimbrial of the fornix. And as you go through these, if you go, where did this come from? Where did this go? What's the purpose of it? Look it up. Have a good anatomy resource. Mostly it's online. I put this picture in because, again, it gives you a sense of 3 dimensions. These are your, the optic radiations. You could see how this would give rise to Meyer's loop down into the temporal lobe and that portion which goes straight back through the parietal lobe. And again, Lots of things being labeled for you. Commiserable pathways from one hemisphere to the other is obviously the corpus callosum, and we showed you the anterior commissure. In general, so of course the corpus can be divided up. When you look at it in the sagittal plane, you can see the rostrum, the genu, the body, the isthmus, and the splenium. In addition, at the genu and the splenium, there's a forceps, the forks, where you can go both anterior and posterior, divide as they go out into the frontal lobes or into the occipital lobe. Of course, now the interhemispheric fissure's in the way, right? And the main function of the corpus callosum is simply to connect virtually every piece of one cerebral hemisphere to its mirror image on the other side. I mean, it's more than that because, as we'll get into detail later in the course, it spreads out a little bit. It's not point to point. It's not point to point, but it's regional to regional, centered on a point to point. So the point to point that's a center, but it doesn't adhere to that strict single point to point. The anterior commissure, again, is interconnecting mainly the olfactory structures here and regions of the middle and inferior temporal gyri. This is a very good set of slides, or a set of pictures on this one slide. Here, first of all, you can see rostrum, genu, rostrum, the genu. Then this is all body, and then there's the isthmus, and then the splenium. This one shows you region by region what they're connected to. This one begins to show you how it starts to look in 3 dimensions. This one is an even better map of how it looks in 3 dimensions, and also talks about the forceps, how they come toward you, separate from one another, almost like an aplesia. You know, I think aplesia when I see this, but anyway, there you are. Pictures. Sitting on top of the corpus callosum are some interesting structures. You have the indusium griseum, which is actually a gray matter layer sitting on top of the corpus callosum. It's very interesting. It's olfactory bulb output goes there. It's a gray matter layer. And then you have these 2 white matter layers, the stria of Lancisi. There's a medial one and a lateral one. Both of these are related to the limbic system output. All right, we're in the home stretch now. We're just looking at pictures. Centrum semiovale, centrum, the central, the semi-round, semi-oval, right? The semi-oval center. Right. Basal ganglia, and we're going to have lectures on the basal ganglia. Believe me, we'll cover them in detail. We're not learning them today. We're just looking at how they're situated in the cerebral hemispheres. The putamen, the globus pallidus. The globus pallidus doesn't belong here. It's diencephalic, right? Caudate, telencephalic, putamen. Nucleus accumbens. I'm going to show you that in a minute. This is a good day to learn about the nucleus accumbens and the amygdaloid complex. And then the corpus striatum, we use these non-anatomic terms, if you will. The corpus striatum, where the lenticular striates are, the putamen and globus pallidus and caudate, because they're all lined up. But that's not an anatomic name. And then the lentiform nucleus, and even that is really not an anatomic name. It's a putamen and the globus pallidus together. All right. Here's, again, the Yakovlev. Oh, by the way, who was Halim? Halim was the curator of the Yakovlev collection. And when it was at the National Medicine Museum, something like that, it now sits in its own collection. And Halim added a great deal to the organization and making tools that allow us to appreciate it. Yakovlev had created the slices and studied them, but they would be inaccessible to us without Halim's contribution. So the collection is called the Yakovlev-Halim system. Again, these are the Nissl. Each of their say, this is their labeled 1451. This should also be this is 1452. Neighboring slices that have been stained for myelin or for Nissl. So, and this is where so caudate unstained, there's your Nissl. Putamen, globus pallidus. I think that in many ways the Weigert is more revealing. So because this would look like a single nucleus, you'd have to be pretty sophisticated to see that that's not as dense as this is. But here you can see that it has more white matter. Remember, it also had a lot more glia. That was the area. This is 4-to-1. This is 100-to-1. Again, looking down, and you can see the thalami, thalamic nuclei on that, but we'll come to that. And this is the again putamen and top of the cerebellum. We different level, and we're looking clearly at the third. We're looking at the top of the cerebellum, staying focused on the basal ganglia. We can see that the caudate is a larger structure than just that head that we think about. Here's the putamen, globus pallidus in this particular plane. Now we pick up claustrum. Very good for seeing. This is the internal capsule. This is the external capsule, and this is the extreme capsule. Okay, you will hear all of those terms and use all of those terms. Just looking at pictures, right? This is a different level. This is a good one for picking up the gyrus rectus. Related to the gyrus rectus. Here's the midbrain, of course. Here is the optic tract. All right. We're getting out into things like well, choroid, but this is hippocampus out here. Hippocampus. We're going to get better views when we turn to the coronal plane, as we did right now. Up in the centrum semiovale here. Not much to see in the far frontal lobe. It's all cortex, no basal ganglia up here. As we move further back, however, you can again see the nissle. All right, so nissle stain, how dense, if anything, you want to take away is how very dense the caudate is. All right, like cortex. Look at how dense the cerebral cortex though is. Much more dense than anything deep, much less white matter, much more dense. Now, here's where I want to be. I want to get to this structure right here, which is here. Look what is this? This is the internal capsule. This is the caudate. This is the putamen. Well, look at this. The caudate and the putamen are continuous with one another. They're derived from the same structure. They are divided from each other by the developing internal capsule, which drives a wedge between them. But given the angle that you would have to see in 3 dimensions, as the internal capsule gets smaller, anteriorly, the caudate and the putamen, their fusion is visible. That fusion is the nucleus accumbens. And the nucleus accumbens is particularly important in our understanding of psychiatric disorders and it's also a major site where Alzheimer's is thought to play a major role. Okay, so we will study the nucleus accumbens later, but you're never going to see it better than this. This is the site where the caudate and putamen. Now, caudate and putamen do different things. I'm not saying that they're all an undifferentiated structure. They clearly are not the same structure. Okay, just by neuronal density, they're obviously not the same structure. But we will come to that later. As we move further back, again, a gorgeous view of the putamen, and look how separate the globus pallidus is with that separation of the internal striae. Again, a very good look at the external capsule and the extreme capsule. Good views of the internal capsule coming down. All right. And we're getting down into diencephalon down here. We're not going to talk about it today. Up here, the septum, the fornices, right? And the fornices and some of the septal nuclei. The caudate reaching out across the putamen to the putamen through the internal capsule. I mean, really nice anatomy with the Weigert. And as we move further back, you're now getting into thalamus, but before we leave, we're now looking at further posteriorly in the putamen and the globus pallidus, and ever more than ever, you can now see the separation of the pars internal from pars external of the globus pallidus. All right. Now you're getting into hippocampus. Notice how far posteriorly we have moved before we get into hippocampus. So don't be looking for it up at the frontal poles where the temporal pole, it's a little bit further back. And you can see the hippocampus here and the dentate gyrus here, all right? And lots of thalami. And you can see how the thalamus is diencephalic. We're not going to talk about it much today, but we will go due attention to the thalamus, I promise you, with lots of discussion of its nuclei. And the other thing that I really like about this is that this is a coronal plane. Look at the substantia nigra. See, I think when you use the traditional axial plane through the midbrain, you think of the substantia nigra as almost like a finger-like projection that you're cutting through. It's not. It's a long sheet that runs the entire length of the midbrain. It's a very long structure, really. It has considerable longitudinal extent, much more than you see. But here you see some of that longitudinal extent. And more importantly, I think, you see this lining up, okay, of all of the subthalamic nucleus and things like that, that now create the substantia nigra, the subthalamic, and how they're all lined up for their motor activities. And so I think it's important to spend a couple minutes and just contemplate the three dimensions of these diencephalic structures. And as we move further back, again, now you can see, look at this just here for a minute, and look at this structure, and you'll see why it's called the seahorse, hippocampus, right? Because it looks like a seahorse. That's how the seahorse lives, okay? That is what a seahorse looks like. So the hippocampus, and strictly back into thalami now, and picking up this, the brain. And then sagittal view and cingulate, that sort of thing. Corpus callosum, good view, of course, of the corpus callosum. And centrum semiovalve. Putamen. Watch what happens to the putamen. Watch this. Look at this. I think this should have been called the shrimp nucleus myself. Every time I see this view, it's like, that is a shrimp, okay? A crawdad maybe, but no, that's a nice big Gulf shrimp. That's how I think of it. Shows where my head is. Anyway, a little bit of a view of the putamen into the thalamus. Thalamus is very, very large. And look at this view of the putamen. Always peculiar, but you always see it like that. And look at this is a good sagittal view of the hippocampus here. Again, a very large structure. Very large, very long. You have to see it in 3 dimensions. That's it. Okay. Next week, another just a gross view of the posterior fossa. All right, and then we'll start getting into some regional anatomy looking about.